weird kind of phrasing questions.
Yeah, that happens. With especially like Ferreira's questions, they can be a little bit confusing. So I'm sorry for that. Hopefully you guys maybe got some points back if you argued them. Because of the weird wording, sometimes we were able to get our points back for that.
You emailed her and she's like, I'm not discussing anything, study ahead of time, take advantage of TA sessions. Like, she's not having a conversation about it.
Really? Wow.
Can't tell anyway. I haven't emailed Dr. Ferreira.
Wow. Okay. Well, I'm sorry about that. Hopefully, I mean, this upcoming test, it's going to be a lot easier. I know Dr. Safang is very intimidating, especially with his Arnold Schwarzenegger voice and his enjoyment with embarrassing people. I know last year he humiliated this kid who just asked like a really general question, but yeah, so be wary of the questions you ask him and definitely turn your mic off.
It's just a thing he does. Like that's just a thing. Because he did that to a girl in our class yesterday, I think.
Yeah, Sarah. She left her mic on. She's actually a good friend of mine, and she laughed it off, so she's fine. But yeah, that was humiliating, and I felt bad about that. But yeah, that's the thing about him. Like, he is very intimidating. But his test questions are very easy and straightforward. So usually, although he's like a rough person to talk to, his tests are like really straightforward. So you've got that to look forward to. He does a lot of talking during his lectures about like random things. And between like me and like the 100 of you guys, like I always zoned out during his lectures because there was just so much. And I did fine. Like I think I got a 98 on that test. So obviously, you know, he likes the big picture ideas and he only tests on what's on the slide. So if you understand the slide and the general concepts, you should be fine. Am I showing the PowerPoint? I thought I was. I don't know if it's going to work because I've been having issues with like sharing my PowerPoint, so I might just have to share my screen. You can see it?
Yeah, maybe it's like a log out, log back in kind of glitch.
Okay. Yeah, try doing that, Kara, if you're not seeing it.
Hey, Phoebe, I also have a question about Dr. Stephen.
Yeah.
So would you say that the things that he says about the PowerPoints are as equally as relevant as like focus exam material or something? Because he says a lot and sometimes it's really hard to interpret exactly what he's trying to say.
Yeah, so I'm going to try to break down like the main concepts, but literally he doesn't test about anything he talks about. Like literally, like I said, I zoned out for most of his lectures and I did fine. You just have to know what's on the slide, and he's not going to ask you on like anything. Like I know he talked a lot about like brown fat and like thermogenesis, but like you don't have to know anything about that. We don't learn that at all. Just kind of like memorizing what's on the slides, knowing the pathways, knowing like the main enzymes. Like when he says he wants you just to know like the main concepts, he means it. I know my year I overstudied for this test because I didn't really trust him not to ask hard questions, but like really I got to the test and I was like very disappointed that I wasted so much time just memorizing literally everything. So you don't have to worry about that. He is going to be one of the easier testers. So that's good. Yeah, so don't worry about the tangents. Don't worry about like, you know, anything that he says. Just worry about the slide material. What he says is just explaining what's on the slide and then like extra material that is really just for your interest. So don't worry about that. I know it's like stressful because you're trying to like write down everything he's saying. So don't worry about that. But yeah, any other questions about that?
Is rewatching his lectures helpful at all?
I literally never rewatched his lectures last year. I think, you know, I just benefited by going to the TA sessions because they were able to like kind of break down the important stuff. So yeah, it's just too much to like rewatch all of that and go through all of his like long tangents of.
Okay, thank you, because it was taking me way too long to get through his lectures.
Yeah.
Okay, thank you.
Yeah, don't worry about that.
He shows in class. He showed like 2 videos, and like one of them there had no sound, and the other one it was like super retarded basic. Like, do we need to watch those again?
No, he's not going to test on any of the videos he shows. Literally, what's on the slide is what's important. And I know he has notes underneath his slide. Don't even worry about those because they're very wordy and he's not going to ask anything about that. That's literally just for you to better understand the material.
Yeah, he was playing one of the videos and he was like, can everyone online hear? And someone answered, they're like, no, we don't hear any sound. And then he just didn't reply and he just kept playing the video. He's like, well, I can hear it. You know?
Yeah.
The class in the building, so.
Yeah, he's kind of rude about that.
I'll be honest with you, Phoebe. I'm one of those people that I've never really missed a class or I've never not gone. Well, of course, after like, you know, with my freshman year aside. And I just stopped watching his lecture after like that last one. It just doesn't do anything for me. I feel like it makes me dumber.
Yeah, no, that's fair.
Yeah, honestly, I feel like it's not doing anything. Are there any other like outside sources that you think are really good for basically like this part of metabolism, like on YouTube?
Yeah, I'll try to post some videos for that. I found a few like over the past few days. I just haven't written them down or like posted them, so I'll try to post them for you.
Okay.
But otherwise, like literally, like I said, I zoned out during his classes, and then I just went to the TA sessions, and that was like enough for me.
Cool. All right, thank you.
You're welcome.
And to follow up, so he's like our only professor for this block?
I think so. I think maybe Patel maybe does one. I don't remember though. But he does the majority of it. But his test questions are easy, so it's not terrible. Just, you know, it's just the lectures that are hard to get through.
Okay, thank you.
You're welcome.
Phoebe, it seems like there is an issue with the slides. Like multiple people have logged in and logged out. Can you maybe like take that back up and maybe that'll fix the issue for everyone?
Yeah, I'm just going to share my screen, honestly, because it took like way too long to like load it. Now you're just looking at my screen, right?
Yeah.
Okay. That works. I'll just share it this way. You won't be able to see my cursor as well, which is kind of sucky, but I'll try to like actually draw everything out so you guys can see what I'm pointing at. But yeah, okay. So the next thing I've gotten a lot of emails about is like how to study for this material. So I already posted some examples of my one sheet slides, which is really just going through each of the slide material and kind of drawing it out as like a whole thing. And I recommend doing that after each class. So that way you guys get like the general like basic concept and how everything connects. I don't recommend flashcards. I know it's like heavy on memorization, but a lot of his material, he likes you kind of like knowing how things fit together. So if you're just memorizing things, you're not like seeing like the whole big picture. So that's why I recommend like just memorizing like the whole pathway and seeing it like as a whole. What else? Okay, so on weekends, I literally went to the GLs every single weekend, and I'd like use the whiteboard space to like write out everything, all the pathways, all the major enzymes, and like if you do it over and over and over again, you can like memorize it essentially to the point where you could just like say it out loud without looking. So that's kind of what I did for memorizing the pathways. It's just a ton of memorization, which is very time consuming, but if you keep up with it, it's not terrible. I usually just like picked up the main concepts first, memorized those, and then went back in and memorized the smaller details as you got closer to the test, because you don't want to like memorize all the small details at the beginning and then forget them when it comes to the test. So I recommend doing that. Other than that, he does recommend making like a chart. So the chart is basically just like he gives you like these like 6 categories, and you have to like compare like the different pathways. I'll show you what I mean by that because he has like the list on one of these slides. I don't remember which one, but I'll show you what I mean when we get to that slide. Yeah. So any other questions about any of that? I don't think I'm like looking at the chat. I don't know. My chat isn't loading. So if I miss your question, can somebody like, you know, read it out for me from the chat? Because for whatever reason, it's just not working.
What lectures are you going to be going over today?
Okay, so I'm going over all 3 lectures. So it might go a little bit over time, but I figured I'd like get it over sooner. So that way we don't have to like try to catch up later.
Yeah. I was just wondering.
Yeah. Okay.
Thank you.
Yeah, no problem. So if you guys can't stay for the whole thing, like that's okay. So we might go over a little bit of time. With that being said, I'm probably just going to start unless somebody has like one last question beforehand. No? Okay. Okay, can everybody see my slide?
Yes.
Awesome, thank you. That's so weird. Okay. Okay, so this first lecture, it is like not terrible, I don't think, but I'm gonna kind of highlight the things he does want you to know. So you do have to know the difference between hexose and pentose. So knowing that it's 6 carbons versus 5 carbons, and kind of understanding that glucose, fructose, mannose, and galactose are examples of a hexose sugar versus ribose, which we've already talked about, is an example of a pentose sugar. And then, you know, I kind of like showed you the difference between like the deoxyribose and the ribose. It's like this 2' carbon has the alcohol group versus just a hydrogen for the deoxyribose, but you guys probably already know that from genetics. I would know the difference between the alpha and the beta. You don't have to know the structures, so don't worry about like memorizing the structures of these sugars. All you have to know is that a hexose has 6 carbons, a pentose has 5 carbons, and then you've got these alpha and beta stereoisomers. So your alpha is going to be pointing down, whereas your beta is going to be pointing upwards. And the alpha is more important for us because that's the one we can break down as mammals. So this is an example of a D glucose, and he'll talk about D and L glucoses in the next slide.
Hey, Phoebe, quick question.
Yeah.
That as far as the alpha and the beta, he just wanted us to know one is pointing down and one is pointing up, because alpha and beta linkages, alpha linkage, if I remember correctly, was like cis, as in both of those oxygens are down, and beta, it was trans, as in one oxygen was up and one oxygen was down. So he just wants us to know one hydroxyl group is up versus down, not the linkage aspect?
Yeah, don't worry about the linkage. We'll talk about that more when we talk about glycogen, so don't worry about it now. But for like this like lecture just by itself, just, you know, knowing that alpha is like the one that we have as like we can break down is like probably the most important aspect. So don't really worry about the actual structural component of it.
Thank you.
Okay. Yeah, no problem. Okay, so he does want to like, you know, introduce the idea of these enantiomers. We've got D sugars and L sugars. The most important one for us, and he has it here in blue, is to know that mammals have, you know, use D sugars, and, you know, we have L sugars in plants and microorganisms, but that's like less important, and we can't digest those. So what we can digest are the D sugars, and only if they're alpha linked, not beta linked. So that's the main point here. You will need to know the difference between disaccharides and polysaccharides, but that's super easy. You know, disaccharides are smaller, so they're better for storage, whereas polysaccharides are larger, so they can also work as like a structural kind of thing. You will need to know the difference between lactose and sucrose. So lactose is glucose and a galactose group, so that's really easy to remember because you have the word lactose right in it. But then sucrose is going to be glucose and fructose, so know what each of these is composed of. Polysaccharides, you would have to know that they're structural. And the most important one for us is glycogen. I don't remember if he actually tests on starch and dextrin because it's like not really relevant. But I would kind of have an idea as to, you know, what starch and dextrin are. You would have to know that glycogen is just going to be a bunch of glucose molecules that are alpha linked to each other. And, you know, that's how you form like the storage for glucose in your body. So that's super important. What's a little bit less important to know is the, you know, the difference between cellulose, glycogen, and chitin. So you have your glucose here, but this is going to be beta linked, and this is found in plants. We can't digest it because it's beta linked. So know the differences between your glycogen and your cellulose, and then also know the difference between your chitin and all of that. So this is your N-acetylglucosamine. I remembered all of these like blue colored, you know, bolded words for my year. So I would suggest memorizing it, but I would say that if anything, you do need to know what glycogen is. You know, you need to know that it's alpha linked and that it's a bunch of glucoses linked together. So read the objectives to kind of like test yourself on your understanding, because he does like the summary slides, and the objectives are like big picture concepts that he's going to ask about. So glycolysis is pretty much taking glucose, turning it into 2 3-carbon compounds and forming ATP from it.
Wait, quick question. Do we need to know the anomeric linkage for the disaccharides? Question from Broke.
Wait, sorry, ask that again.
Do we need to know the anomeric linkage? I'm guessing that means like the number for the disaccharides, like as in it's galactose and glucose equals lactose?
Oh, yeah. So knowing what lactose is composed of and what sucrose is composed of that's important, yeah.
Thank you.
You're welcome. Okay, so also you have to know, is there a question?
Yeah, the N-acetylglucosamine, that's just part of chitin?
Yeah, so instead of having a bunch of glucoses linked together for chitin, you're going to have a bunch of N-acetylglucosamines beta linked to each other.
Gotcha. So there's no glucose, it's just the N-acetylglucosamine.
Yeah, correct.
Okay, thank you.
Sorry. Okay. So you do have to know that glycolysis happens in the cytosol. In fact, you're going to have to know where each of these processes occur. So know that glycolysis is in the cytosol, know that your electron transport chain is in the inner mitochondrial membrane, etc. So that's super important. Okay, so essentially glycolysis is a sequence of 10 reactions that converts glucose into pyruvate. So you start off by using 2 molecules of ATP to start off this reaction, ultimately making a total of 4 ATP. But since you are using 2 to make 4, your net production is only going to be 2 ATP. And then you also have to know what the oxidative step is. And we talk about that in a few slides, but knowing that you use this NAD plus to kind of take a hydrogen from the molecule, and that allows it to convert it to the next product. So like he said, don't memorize all the enzymes. I did, and I regretted it. So seriously, don't memorize all the enzymes. A kind of thing, like a common theme to know what to memorize and what to not memorize is if he has a slide on an actual part of the reaction. So if he focuses on something or zooms in on a particular reaction, he wants you to know it. And it's also important to know what the rate limiting step is, which in this case is phosphofructokinase. So that's different between your normal cells. So all other cells have the rate limiting step of phosphofructokinase versus your beta pancreatic cells, whose rate limiting step is glucokinase, which is your isomer of hexokinase. So he just wanted to give you this big picture kind of how everything works together to show you what happens. I would know hexokinase as the first step. It's also, a step that uses ATP, so that's kind of high yield. I would also obviously know the rate limiting step, which also uses ATP, so know that and know these, reactants in the product of this reaction. So know glucose to glucose 6-phosphate. Don't really worry about this isomerase. It's lower yield. And then knowing that your phosphofructokinase is going to phosphorylate carbons 1 and 6, and that's going to kind of destabilize it, so it's more likely to break into 2 pieces through aldolase, which breaks it into your glyceraldehyde 3-phosphate and your dihydroxyacetone phosphate. So your glyceraldehyde 3-phosphate can proceed down the rest of glycolysis and yield 2 ATP. So I would know this step here because it yields ATP, and I would know this step here because it also yields ATP. So I would say those are the high yield steps. After that, you get 2 ATP from that. You're left with your dihydroxyacetone phosphate. It's going to go through the isomerase to become glyceraldehyde 3-phosphate, and it's going to go through the rest of the process as well. So then you get 2 more ATP for a total of 4. And then you're only making a net 2 because you used 2 to make the fructose 1,6-bisphosphate. Okay, so here he's kind of introducing the idea of your hexokinase and your glucokinase. So I hope you guys remember when we talked about isoforms, how they're enzymes that do the same reaction but are found in different tissues. So hexokinase and glucokinase are examples of isoforms. So hexokinase is found in all of your normal cells, and glucokinase is found in your liver and your beta pancreatic cells, but we talk about it specifically in your beta pancreatic cells, so don't worry about like knowing that it's in the liver too. So here we have your alpha D glucose, like we talked about, your alpha stereoisomer, your D enantiomer, and then, you know, it's glucose. So this is the one that we use. We go through either hexokinase or glucokinase to make your glucose 6-phosphate. This is a lower yield enzyme, so I wouldn't really worry too much about memorizing it. This is your rate limiting step, at least for your normal cells. So memorize that. You use ATP to break it down, and we talked about this already, so I'm not going to go over it again. But from this slide, know this, and then have a general idea that you're going to have this aldolase form 2 3-carbon products. So you go from 6 carbons to 3 carbons, or 2 3-carbon products.
Phoebe, is your pointer on? Because I can't see what you're pointing to when you say, like, know this kind of thing.
Oh, I'm sorry. Like, I'm sharing my screen. No, I think it's because I'm sharing my screen, because like usually if I upload the PowerPoint onto it, it would like show the pointer, but it wasn't working, so I'm trying. Okay, I'll just circle everything that I'm like talking about.
I'm sorry.
No, I'm sorry. Okay. Okay, so yeah, you're gonna get your 2 3-carbon compounds that will then undergo glycolysis. Okay, so this is a super important step to understand. It's going to be your oxidation step. So what's important here is having this NAD plus to grab the hydrogen from your glyceraldehyde 3-phosphate to convert it into your, wait, which direction? Oh, I'm sorry. It takes the glyceraldehyde 3-phosphate, takes the hydrogen from it to turn it into your 1,3-bisphosphate glycerate. So I would know this reactant, this product, and it being the oxidative step or oxidation step. So what happens here is that you need to replenish this NAD+, and that can be done in 2 ways. So in the electron transport chain, we use NADH as like a source of energy, essentially. So you can, if you have oxygen available, then you can go down the electron transport chain and, you know, use up that NADH, convert it back to an NAD+, and bring it back for glycolysis. But under conditions of low oxygen, you're going to have to undergo anaerobic respiration, and then that's going to rely on this lactate dehydrogenase, which is converting pyruvate to lactate. And this is not ideal because lactate is acidic, and that's not good because it will lower your blood pH. So ideally, you want to be, you want it to have oxygen to undergo the respiratory chain, which is also going to give you like 36 ATP. So it's also ideal for energy and most efficient. So pretty much everything I circled is important to know. I would definitely know lactate dehydrogenase as an enzyme for converting pyruvate to lactate, and also replenishing the NAD+ to give it back to the oxidative step. So for that reason, I would probably know this enzyme somewhat. It's a little bit lower yield, but kind of be familiar with this enzyme and how it's, the oxidative step. But lactate dehydrogenase is probably more important.
Hey, Phoebe, do you mind going to that slide? I just want to make sure it's clear for me. So this 1,3-bisphosphoglycerate can either go through the respiratory chain, or it can go and make lactate dehydrogenase, and that's just dependent on oxygen, right?
Right, yeah. Okay, so the idea is it's the NAD+. Like how you get your NAD+ is how, you can, whether it goes through the lactate dehydrogenase or the mitochondrial respiratory chain. So it goes from G3P, so this enzyme here, to 1,3-BPG. So it's moving in this direction. Sorry, it's like really, I tried to number it. So your glyceraldehyde 3-phosphate, I'm going to clear this slide. Sorry. So your glyceraldehyde 3-phosphate needs NAD+, because this NAD+ is going to remove the hydrogen to convert it into 1,3-biphosphoglycerate, right? So you need to replenish the NAD+ so that it can do this. So the way you can do that is either take this NADH, put it through the electron transport chain, which, takes off the hydrogen and, gives it back as an NAD+. If there is oxygen present. If there is no oxygen present, then you can't, do any kind of mitochondrial stuff because that is dependent on oxygen. So it's going to have to go to lactate dehydrogenase as an NADH. The lactate dehydrogenase is going to remove the hydrogen group and give it to the pyruvate to convert it into lactate. And so now your NAD+ is back to being an NAD+ and can go back and, work its role in the oxidative step. Does that make sense?
It does. Thank you.
You're welcome. Okay, so I would also know what steps undergo substrate level phosphorylation. So substrate level phosphorylation is really just when this substrate, which is this thing right here, gives the phosphate group from itself to the ADP to make it into ATP. And the enzyme that kind of helps this out is going to be the phosphoglycerate kinase. So I would know this enzyme as being involved in substrate level phosphorylation. I would also know, these two substrates and products that allow for this. And also phosphoglycerate kinase, we talk about its like regulation. So that's also important to know. And then also another enzyme involved in substrate level phosphorylation is this your pyruvate kinase. So this is your phosphoenol pyruvate, or you may know it as PEP. It's going to give its phosphate group to the ADP to turn it into ATP. So this is also an example of substrate level phosphorylation. So no pyruvate kinase and phosphoglycerate kinase as your two substrate level phosphorylations. And they're both kinases, so that shouldn't be too difficult to remember.
I thought it was pyruvate kinase and PFK1 for glycolysis.
Wait, say that again?
Phosphofructokinase, not phosphoglycerate kinase.
As a substrate level phosphorylation?
Oh, this slide is substrate level phosphorylation, but the regulation is phosphofructo or PFK.
Yeah. That's the rate limiting step, but pyruvate kinase is also an enzyme that is being regulated. It's just not the rate limiting step. So we talk about both of them getting regulated. Sorry if I said something different.
No, that's okay. I just wanted to make sure I was understanding what you're saying correctly so far. Thank you.
Yeah. Okay, so now we're talking about regulation. So you've got your 3 enzymes that are being regulated. So you've got your major control point, which is going to be your rate limiting enzyme, which makes sense. And then you've got your minor regulators, which are also going to be regulated, but not as extensively as your phosphofructokinase. Like I said, you've got your pyruvate kinase, your hexokinase, and your phosphofructokinase, which are regulators. So specifically for your phosphofructokinase, it's going to be regulated by high ATP levels, so that will inhibit it, which makes sense because ATP is a product, and products typically inhibit the enzymes that make it. Also with the proton concentration, lactate is derived from your pyruvate, which is a product. And so if you have a lot of lactate, you're going to want to stop glycolysis because that probably means you have a lot of pyruvate as well. And you're undergoing anaerobic respiration, which is not ideal. So it's also going to go back and inhibit phosphofructokinase, the rate limiting enzyme. And then you'll also learn this later on, but citrate, which is a byproduct of the TCA cycle, it's used in fatty acid synthesis. So if you have a high amount of citrate, it's a byproduct of the TCA cycle. It's being used in fatty acid synthesis. You're going to want to inhibit glycolysis because now you're making fatty acids. So why would you be burning glucose if you're making fatty acids? So high citrate levels or the availability of other kinds of fuels like ketone bodies or fatty acids. Don't worry too much about the details here because we're going to learn it in block 3. And then your insulin to glucagon ratio. So insulin is important because it is what brings glucose into the cells, essentially. So if you have a lot of insulin, that means you're bringing more glucose into the cells. and that's going to want to stimulate glycolysis. So if you have a high blood glucose level, you want to drop that blood glucose by bringing more releasing insulin so that more glucose can enter the cell, and that will stimulate glycolysis. Versus if you have a low blood glucose, you want to bring those blood glucose levels back up. So you're going to release glucagon, which will then increase your blood glucose levels, and therefore you're not going to want to be doing glycolysis. So he gives the example of high fructose 2,6-bisphosphate. This is an important, molecule that you need to know that is a regulator of the phosphofructokinase. So we've learned about fructose 1,6-bisphosphate, right? That is going to be the product of fructose 6-phosphate. So sometimes when fructose 6-phosphate builds up, it can either go through like the normal enzyme, I think phosphofructokinase, sorry. Phosphofructokinase is going to make it into your fructose 1,6-bisphosphate, but when it builds up, this phosphofructokinase cannot keep up, so some of this is going to be shuttled into another enzyme that we don't need to know about, but it makes it into fructose 2,6-bisphosphate, which is, the wrong phosphorylated version of it. So when this builds up, you know that you have a buildup of, this reactant. So you're going to want this fructose 2,6-bisphosphate to come in and upregulate phosphofructokinase so it works faster and can, start, making glycolysis go faster to get rid of this increase in fructose 6-phosphate. So know this as an allosteric regulator, which we'll talk about next. So this is the Michaelis-Menten plot again. You don't have to know this at all, like literally don't even bother with trying to figure this out again because we've already learned it. But pretty much what he wants you to understand is that it's an allosteric regulator, which will increase the activity of phosphofructokinase. And when you have like an allosteric regulator, it increases the relative velocity of the enzyme or the reaction, and then that just shifts the curve to the left. So don't worry about that, but knowing that it's an allosteric regulator or activator is super important. Okay, so now we're talking about the regulation of pyruvate kinase. So this is a different enzyme. This is the one that takes PEP and turns it into pyruvate. Okay, so this is the last step essentially. And it's going to be regulated in two ways. It's got allosteric regulation, which is your fructose 1,6-bisphosphate, not to be confused with your fructose 2,6-bisphosphate, and ATP as well as alanine. So we know that your fructose 1,6-bisphosphate is going to be a reactant. So if you increase the amount of reactants, you're going to want to increase the speed of glycolysis so that it can keep up with the amount of reactants available. So obviously fructose 1,6-bisphosphate is going to upregulate the pyruvate kinase. ATP and alanine are both going to essentially be products. So remember, I don't know if we got to that slide. No, we haven't gotten to that slide yet where we talked about how pyruvate is turned into alanine so that it can, travel to your liver. So essentially, when you have high amounts of pyruvate, you're going to have a high amount of alanine in the blood. And as a product, it's going to want to inhibit the enzyme that makes it. Same thing with ATP, which is also a product. It's going to inhibit the enzyme that makes it. So know these as allosteric regulators. On the other hand, the amount of blood glucose that you have in your blood is also going to regulate it, but in a more covalent kind of way where it, phosphorylates or dephosphorylates the pyruvate kinase. So typically, if you're dephosphorylated, it means you're more active, whereas phosphorylated means you're less active. So if you have a low blood glucose level, you're not going to want to bring glucose into the cells, and you're definitely not going to want to do glycolysis. So what this does is it will activate phosphorylation of your pyruvate kinase and inactivate the dephosphorylation of pyruvate kinase so that more of it is going to be in the inactive version. On the contrary, if you have high blood glucose, you're going to want to inhibit the phosphorylation because now you're going to want to initiate glycolysis. So inhibit the phosphorylation and upregulate the dephosphorylation so that you have more pyruvate dephosphorylated, making it more active. So read through his summary slides because he's going to test on these, especially if he has them bolded or in blue. Like, that's important. Yeah. So for insulin secretion, so this is only in your beta pancreatic cells that will be releasing insulin. You have a different rate controlling step. So in your normal cells, your rate limiting step is going to be your phosphofructokinase. But in your beta pancreatic cells, you're going to have your rate limiting step as your glucokinase, which is, the different version of your hexokinase. So essentially what glucokinase does is it is going to take glucose, phosphorylate it so that it can't leave the cell. So it's stuck in the cell. What this also does is that it changes glucose's identity such that now the cell looks like it has a low concentration of glucose. And so that allows more glucose to flow down its concentration gradient into the cell. So if you have a glucokinase that does not work properly, you're not going to be able to get as much glucose into the cell. And as a result, you're not going to be able to initiate glycolysis and then the following steps that result in insulin release. So some type 2 diabetics have this mutation in the glucokinase gene such that it doesn't work properly, and then therefore they can't release insulin or as much insulin. I would memorize the GLUT2 transporter as being the important transporter for glucose in the beta pancreatic cells. I think it's called GLUT1 in the other cells, but don't worry about that. I would know it as a low affinity, high capacity, which basically means that you have to have a pretty high blood glucose concentration for it to allow glucose to enter. And the reason for that is that it needs to be very specific. Because if you let glucose enter the beta pancreatic cells, it's going to want to release insulin. So it needs to be very specific to, ensure that you're not, secreting too much or too little insulin. So your pancreas has these little areas on it, like little circles of cells that congregate, known as the islets of Langerhans. And they're composed of several different types of cells, but the two that you need to know are your alpha cells, which secrete glucagon, which we'll talk about later. And then the most important for this lecture is your beta cell, which is going to secrete insulin. And pretty much you have to know this whole reaction, how everything works together. So it starts off by these beta pancreatic cells sensing an increase in your blood glucose level. And then your GLUT2 is going to sense this, become more sensitive to allowing glucose to enter the cell. And once glucose enters the cell, now your glucokinase can phosphorylate glucose, and that initiates glycolysis, but also promotes more glucose for entering the cell. So the next step is after it goes through all of glycolysis, it's going to make pyruvate. Pyruvate will enter the TCA cycle, which would then enter the respiratory chain, which makes a ton of ATP. So now your concentration of ATP to ADP is going to increase. So you're going to have a ton of ATP. So when you have a certain concentration of ATP, the ATP is going to come into this potassium channel and block it so that now it is closed, and then potassium can no longer leave the cell, and you wind up with membrane depolarization. So when the membranes depolarize, this voltage-dependent calcium channel will allow calcium to enter the cell, and when calcium enters the cell, you can release insulin. It triggers the release of insulin. So it's a very specific process and only reacts to a certain amount of blood glucose levels for the release of insulin. So this is basically the same thing all over again, but what he wanted you to know about this slide is that these drugs that you need to memorize, your sulfonylurea and your repaglinide, are going to block this potassium channel so that it will allow for the depolarization of the membrane. So if your, say your glucokinase isn't working, or for whatever reason, you're just not allowing glucose in the cell, or it's just not working properly, and that's the case with people with type 2 diabetes, you can just trigger the depolarization, which will ultimately release insulin without having to go through glycolysis or, having to worry about getting enough ATP to trigger this. So definitely memorize those drugs. Okay, and definitely read the summary slides as well. So this is lower yield because you're going to learn about this in greater detail when we talk about it in physio, but pretty much you have a pre-absorptive phase. So you see food, your brain sees that you're about to eat, activates the vagus nerve, which is your parasympathetic, and that's going to tell your pancreas to start releasing insulin. Then you've got your absorptive phase. So after you eat and you start digesting food, it's going to tell your brain that you're digesting food, and then your brain does the same exact thing where it just releases acetylcholine. So from this, I would understand that acetylcholine will stimulate insulin secretion after eating and before eating. As for this whole thing here, like that's super low yield because it's definitely more physio than anything else. Okay, so this is what I was talking about earlier about how you have your Cori cycle, which is less ideal because for your red blood cells or your muscle cells, to kind of like move this carbon skeleton and recycle it, which is what happens with like a lot of things, like you recycle a lot in your body, you're going to, maybe undergo anaerobic respiration. You'll have an increase in lactate. The lactate can then, enter your blood and go to your liver, but this is bad because it's going to be, make your blood more acidic. So less ideal. So know that the Cori cycle, is shuttling lactate from red blood cells, muscle cells to your liver. And then your alanine cycle, which is a little bit better. Does someone have a question? I think somebody's mic is on. Okay. So your alanine cycle, which shuttles alanine to from your muscle to your liver, is like a little bit better because alanine is a neutral amino acid. It's not going to change your blood pH. So by converting pyruvate into alanine, you can move it without changing your blood pH. So it's a little bit better. We'll talk about this like at least 2 more times in the other lectures. So if you don't understand it now, we're going to talk about it again. So no worries. Okay, and then your diseases. So you do have to know the diseases, specifically your pyruvate kinase deficiency. So what happens here?
Did you change slides? We're still seeing Cori cycle and alanine cycle.
Oh, really? Did it freeze? Can you see it now?
I see the summary of insulin secretion slide. Is that what you're on?
I was on it. I just switched it over.
Yeah, diseases.
Okay, you can see it now?
Lag is all.
All right. Sorry about that.
Oh,
it's probably my internet. Okay, so your pyruvate kinase deficiency, literally the only thing you have to know about this is that your pyruvate kinase, your enzyme, is going to be deficient. So you have to associate it with the idea of spiny red blood cells or echinocytes, because he's going to like literally use either spiny red blood cells or echinocytes as like a key phrase for having you determine what kind of hemolytic anemia it causes. Because you're going to learn about another enzyme that, also causes hemolytic anemia. So knowing the difference between, I guess, it's the glucose 6-phosphate dehydrogenase deficiency versus your pyruvate kinase deficiency, your keyword is spiny red blood cells or echinocytes. For the cancer, pretty much, that cancer has a high metabolic rate. So as a result, it's going to take up a lot of glucose. So this is useful for like PET scans to find, areas of like tumor cells, because your PET scans will use this as a like radioactive like sugar that will be taken up by cells that have a high metabolic rate. And because they're radioactive, you can see it in the PET scan. For cancer, just kind of knowing that it has a high metabolic rate and that, you can use PET scans to kind of see this is enough. As for like this, I probably wouldn't memorize it. It's kind of low yield. So yeah, that's, I think, the end of that lecture. Do you guys have any questions about the glycolysis lecture? No? Okay. I'm going to go on to the next lecture. Let me know if you guys can't see my screen.
We can
Okay. Okay, thank you. Awesome. All right. So for the TCA cycle, you don't really have to know all of the enzymes and you don't have to know all like the products. So kind of like glycolysis, you don't have to know everything, just like the things that he like focuses on. So you have to know that the TCA cycle, you're gonna see a lot of it in. Okay, you're gonna see it as kind of like connected to a bunch of other stuff that we're gonna learn about as this semester progresses, because it's connected to so many things. So it's like not super important to know all of the key steps now because it's all gonna like come together later on. But because he kind of repeats this several times in his slide, I would memorize these numbers because you see it like 2 more times where it's most cells have 500 to 2,000 mitochondria, and then you don't have a nucleus or mitochondria in your erythrocytes because they get rid of them before they mature. And so I'm sorry if you guys hear the lawnmower, guys. It's kind of loud. But anyways.
That's funny. I thought it was at mine because I have lawnmower guys at my place too.
Oh, really?
I thought it was me.
Okay, so if you so pretty much you have to know that the TCA cycle is happening in your mitochondrial matrix. It's going to produce ATP through aerobic respiration, meaning it needs oxygen to function. So anything that's going on in the mitochondria needs oxygen. It's also going to be, create a common product, which is acetyl-CoA, and you're going to see this a few times. We talk about it with, glycolysis, and then we also talk about it with beta oxidation, which we'll learn about in block 3. So don't worry about that too much now. And then, the TCA cycle is an amphibolic pathway, which means it does catabolism and anabolism. Okay, so from this slide, the main takeaway here is kind of being able to see the changes in the amount of carbons for each of these. So your acetyl-CoA, which is going to be derived from your pyruvate, only has 2 carbons. So it combines with its oxaloacetate, which has 4 carbons, and together they make a 6-carbon compound called citrate. Of that, I think that's the most important part of knowing, like, whether you have 2 carbons, 4 carbons, like just knowing the amount of carbons for these 3 molecules is probably the most important. But you can also see that you have your succinyl-CoA and your acetyl-CoA as being your energy-rich molecules, which he talks about a few times, and your products, which, you got like your 3 NADHs, your 1 FADH, and then your 1 GDP to GTP, kind of understanding how that, where they come from. So, I'll get into that a little bit more.
Phoebe, for like the TCA cycle, do we need to know each step that NADH and FADH2 is like added to, or like I guess reduced in? Because he seemed to like harp on that, and I'm not sure if he just wants us to know like 3 of them or if he wants us to know the specifics.
Yeah, so unfortunately you do have to know which steps yield NADH and FADH and GTP. Those are like the most important steps for the TCA cycle. So while you don't have to memorize like all of the steps, I would know which enzymes are associated with, the formation of NADH or FADH. And yeah, sorry, it is a little bit.
Gotcha. I just wanted to make sure. Thank you.
Yeah, no problem. Okay, he like posted this slide again in the electron transport chain, but pretty much the main point here is that, your mitochondria is important for housing the location of the TCA cycle, which is in the matrix here. Beta oxidation, which literally just memorize that because we're going to learn about fatty acids later on. And your oxidative phosphorylation, which is your electron transport chain, which is in the inner mitochondrial membrane, so somewhere in here. And then, he shows this slide again. It's like an intro to your electron transport chain. Not a big deal. Read the objectives. Okay, so when I was talking about making a chart for all of the pathways, this is what I mean. So if you were to make a chart and like, on Excel or whatever, and you put like glycolysis here, TCA cycle here, and then you made like all of these like columns, like the first column would be like the function in the cell or the body, and you categorize everything. That way you can see how they all compare and contrast to each other. And I think I did that my year, so I find it super helpful to do that. So yeah, he recommended it our year. I don't remember if he said anything this year, but definitely try doing something like that, so like you can compare everything and see it as like a whole. Okay, so that's the point of that slide. So he uses this slide again, like later on. So I would say it's somewhat high yield because he uses it so many times. So pretty much the bolded stuff is what he wants you to know. So essentially, your mitochondria has these things called cristae, which are just infolds of the inner mitochondrial membrane. And the reason for that is to increase the surface area so that you can have all of these electron transfer systems all fit in there, because you have like over 10,000 of them. So you need a lot of surface area to fit it all. Yeah. Other than that, he repeats the slide later, so it's like not super important to go over right now. Anyways, so you have your glucose, which then ultimately goes through glycolysis and makes pyruvate. And pyruvate is a super important like intermediate molecule because it kind of gives the carbon skeleton to, to form alanine, which we kind of already talked about with the alanine cycle, oxaloacetate, acetyl-CoA, which we talk about in the TCA cycle, and then also with lactate, which we kind of already talked about. As well. So I've kind of like, these are the enzymes, I've color coded them to their, respective like reactions. So like which one's which. I would probably memorize this because we do kind of talk about these things. At least for like the alanine, I think it's maybe a little less important for now because we will talk about like amino acid synthesis, and we'll actually talk about this in greater detail, but we've already learned about these three in pretty good detail. So I would at least memorize those. Okay, he talked forever on this slide, but the main point that he wants you to know is the fact that you have a porin, which is going to allow for pyruvate to enter the mitochondria. So remember, pyruvate comes from glycolysis. Glycolysis is in the cytosol, and so your pyruvate is going to come through the porin, which is a less selective channel, and then it's going to enter the intermitochondrial space, And so from there, it needs to get into the actual mitochondrial matrix. So it has to go through the second membrane, which is the intermitochondrial membrane. And this membrane is also housing the electron transport chain. And so it's very important to maintain the proper charge difference, because, the proton concentration changes the charge, and that's what helps with the movement of protons to make ATP. So you have to maintain it, and it's super important. So in order to do this, all of these molecules will kind of move with their respective, like either opposites or same charges to maintain this gradient at an appropriate charge. So pyruvate, this is the main one here. Pyruvate moves in with a proton because pyruvate has a negative charge. Proton has a positive charge. Together, it's a neutral charge. So all of these are examples of molecules that need to move in and out of the mitochondria, but they do so in such a way so they don't change the membrane potential, except for ADP and ATP. But the whole point of the electron transport chain is to change the amount of ATP. So if ATP changes the membrane potential, it's not that big of a deal. So I would just memorize this one right here as pyruvate moving in with a proton, and the transporter is called pyruvate symport. So I kind of memorized that my year just because it was the most important one that he talked about. But as for the rest of this, don't memorize. He literally said not to memorize it, and I don't remember memorizing any of this my year except for pyruvate symport. So yeah.
I have a question on this, Phoebe.
Yeah.
So it's definitely not getting in passively. Is it using energy to get in, or is this facilitated? How's it getting in again?
Good question. I honestly couldn't tell you. I feel like, I'll have to look that up. I honestly don't know.
Yeah, I don't know if it's significant. I was just wondering, since we do have to like keep track of like ATP being, produced and used. So I didn't know if this fed into that or not.
Yeah, I'll double check for you because that totally makes sense. I don't remember if he mentioned it. Did anybody catch him mention it?
He didn't. He just harped on Simport.
Okay, so he just didn't say. Yeah, I guess then it's not important.
Okay.
But if you're curious, maybe look it up.
All right, thank you.
You're welcome. Okay, so yeah, this is super important, the pyruvate dehydrogenase complex. You have to know like each of these enzymes that work together to essentially make acetyl-CoA and an NADH molecule. So from my year, I kind of made sure to know the order in which they like work together. So you start off with your pyruvate molecule. It enters the pyruvate dehydrogenase, and that kind of removes a carbon dioxide. So it removes a carbon group from pyruvate, which makes sense because your acetyl-CoA is only going to have 2 carbons. So your pyruvate dehydrogenase will kind of grab it and kind of like, you know, move it in this direction, kind of like, I almost want to say like gears. So your pyruvate is grabbed, moves in this direction. So now the second gear, which is your dihydrolipoyl transacetylase, it has this lipoic acid prosthetic arm, which is super important to know about. And it's, you know, off of a lysine chain. So it will then kind of move in this direction. It grabs the pyruvate from it. And in doing so, it breaks these 2 sulfide bonds. And, you know, and then it moves in this direction. You add your CoA, which your CoA is an energy-rich molecule. It has like, I think 2 or 3 phosphate groups associated with it. So it's like high energy, and it provides energy for this whole, you know, thing. So then you release your acetyl-CoA, and your lipoic arm is still broken. You see these 2 sulfurs they have the hydrogens on them. So they're still broken, and they need to go back and form this ring again. So to do that, now you have your FADH. FADH comes in, and it takes the 2 hydrogens from the sulfurs and, you know, allows it to reform that ring. And in doing so, makes an FADH, which then gives its 2 hydrogen groups to the NAD plus to make an NADH. So when you're counting your like important molecules, like you do have to take into account that this pyruvate dehydrogenase complex, which is something that happens between glycolysis and before the TCA cycle. So after glycolysis, before the TCA cycle, you have this PDH complex working. It makes an NADH. So while your TCA cycle makes it makes 3 NADHs. Your glycolysis is going to make, I think, 1 NADH. But your pyruvate dehydrogenase also makes an NADH, so you have to make sure to count that as a source of NADH. But for the most part, I would understand what a lipoic acid prosthetic group. This is the main enzyme to remember, but I would also know the order in which they occur. All the little details I described to you are less important, but knowing that you make an NADH, an acetyl-CoA, and a pyruvate is converted into acetyl-CoA is like the big concepts here. Okay, so this is also super important. I highlighted all like the key things to remember. So essentially you have a PDH kinase, which is going to phosphorylate this PDH complex that we just talked about. And by phosphorylating it, it turns it off. When you dephosphorylate it through a PDH phosphatase, it's going to remove the phosphate group and it's going to turn it on again. What regulates this is going to be your products, which makes sense. So because PDH is going to be dishing out acetyl-CoA and NADH and also yields ATP, you know, as you go through the TCA cycle and all of that, all of these products are going to go back and upregulate the kinase, which ultimately inhibits the PDH complex, which makes sense. So they don't get it confused with like what's inhibiting what, because there is this middleman PDH kinase that is doing the inhibiting. So you want these products to upregulate the kinase to inhibit the PDH. The other one I would kind of remember is the insulin. Remember when you have insulin, you have high glycolysis. So you're going to want to, you're probably going to have a buildup of pyruvate, and you're going to want to turn on this PDH complex. So in doing so, you want your insulin to upregulate the PDH phosphatase. Why it doesn't do the opposite is, I don't know why, but it just works on PDH phosphatase, and it dephosphorylates the PDH so that it is now active and can promote the TCA cycle, so ultimately. And then pyruvate, if you have like a lot of pyruvate, you're going to want to upregulate the PDH complex to initiate the TCA cycle by making more acetyl-CoA. So your pyruvate is going to come in and inhibit the PDH kinase. Hopefully this was clear, because this is a big key concept that you do have to memorize. So I would memorize the things I've highlighted here. This is literally the exact same thing, so don't worry too much about that. But yeah.
I have a quick question, Phoebe.
Yeah. So
you explained that really well. I just want to double check, because this thing gave me nightmares in undergrad.
Fair enough.
So, the acetyl-CoA, or excuse me, acetyl-CoA. So it will negatively inhibit PDH, but it positively activates the PDH kinase, which negatively inhibits PDH. Is that
yeah. So it's like it activates the kinase to turn off PDH, essentially. Okay. Does that make sense?
Yep, it does. Thank you.
Awesome, no problem.
Hey, Phoebe, I'm glad Lance asked that question, because I was just about to say something about that. I was a little confused because I thought this was also, how we had allosteric and covalent regulation on the other enzyme. This one, I thought acetyl-CoA and NADH, which are products of the PDH complex, went back to allosterically inhibit the PDH complex, in addition to the covalent modifications done by the phosphatase and the kinase. That's what I took from this. That's why I guess I'm a little confused.
Does it do both? Did anyone else hear that? Because it might have done both.
What was it again?
She said that it works as an allosteric regulator and a PDH kinase upregulator, like directly versus indirectly.
Yeah, I think that's what the lines, I think the figure, it says something about squiggly lines versus like just straight lines, like one's a direct.
Oh my God, you're right. You're totally right. Yes, it is an allosteric regulator. Yeah, the squiggly lines are the allosteric regulators, right?
Yes, direct allosteric is squiggly, I think.
Yeah. Yeah, okay, yeah. Arrows with wavy shafts indicate allosteric effects. Okay, yes, so it's both. Thank you for correcting me. So your PDH kinase will, your PDH will, or your acetyl-CoA will allosterically regulate your PDH kinase, which then will phosphorylate the PDH, but also will inhibit directly to the PDH itself instead of going through the kinase. So it does both. Is that right? Am I saying that correctly?
Yes, you're okay. We're all on the same page now, because there was Jillian in the chat also said that she wrote the same thing, that PDH was regulated allosterically via the left stuff and covalently via the stuff in the middle.
Okay. Yeah. Thank you for correcting me. That totally makes sense. Gotcha. All right. Okay, so this is lower yield, but I would kind of understand at least the sources of acetyl-CoA, because we're going to learn about this in detail later on. We already talked about how pyruvate becomes acetyl-CoA, but you also have amino acids and fatty acids that will also make acetyl-CoA. And then that acetyl-CoA can go in and, enter the TCA cycle. It could also help make ketone bodies, and also helps with sterols and fatty acids, making those. But this is the big key concept here. I probably would just memorize this slide for now, because you're going to see it again. So the main point of this slide is just to kind of point out your high energy molecules, which are these ones with the CoA groups, and your succinyl-CoA here, because also has a CoA group. And your CoA groups have those phosphate groups that help kind of energize the cycle to allow it to proceed, essentially. So I have highlighted the enzymes that you should probably know, because these are the ones that are also, taking in these oxidizing agents. So your NAD is going to become your NADH here. It's also going to make an NADH here, your alpha ketoglutarate dehydrogenase. and then your FADH2 as well. And, so knowing the enzymes that make your products are super important as well to memorize. So at the very least, memorize the enzymes that I highlighted here. But other than that, he's just kind of summarizing the whole thing. For the structures, you don't have to know the structures. All this written in red is just kind of like the chemistry of it, which is also low yield. He's not going to ask about the chemistry of it. Okay, so here he called these the energy producing steps because they have a negative delta G value. So they essentially release free energy when they occur and allow the cycle to proceed. So these are going to be your citrate synthase enzyme step, your isocitrate dehydrogenase step, which is also your rate limiting enzyme, and your alpha ketoglutarate dehydrogenase step as well. So I would know these 3 enzymes as being your like energy producing step enzymes. And then, your overall reaction, you can see that it has a very negative value. So it's very favorable as a result. Yeah, so I'm really sorry about this because you do have to kind of memorize, like, at least for, the major enzymes here, knowing what inhibits and which activates each of these is important. Maybe not so much for your pyruvate dehydrogenase complex, but at least for your like citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase complex, knowing, what activates it, what inhibits it for each of these. So you pretty much just summarizes the 3 enzymes that are, activated or inhibited by these things. And essentially you can pretty much summarize that it's going to be the products that will inhibit, and the, if you have more ADP, you want to make more ATP. So ADP will activate the cycle. And then calcium, I don't really know if he explained why calcium does it, but I would just memorize that as well. Does anyone know why calcium does it? No? Okay, well, I guess I would just memorize that.
Phoebe, it's because the muscles, when your muscles contract, you need calcium. So it's
okay,
like,
so it just assumes a higher metabolic rate because you're contracting.
Exactly. So you need ATP. So for the muscles to contract, you need ATP, which needs calcium. So you're making ATP through this pathway. That's why you have calcium as an upregulator. Yeah.
Awesome. Thank you for sharing. Cool. Okay. So yeah, you're going to learn this in greater detail, so I wouldn't worry about it for now. But really, he's just kind of introducing the fact that the TCA cycle has, products that help with other types of metabolic processes. So I think we'll learn about heme synthesis, and we're going to learn about amino acid synthesis later on, as well as gluconeogenesis and fatty acid synthesis. So this is just like a introductory slide that I wouldn't worry about until we get into the details of it. Okay, so here he's talking about how if you were to run out of oxaloacetate, because you need both acetyl-CoA and oxaloacetate to be able to make this citrate here. So if you were to run out of oxaloacetate, for example, there is a way to replenish it, which he talks about here. So usually oxaloacetate is derived from aspartate, and you'll learn this in greater detail when we learn about amino acid synthesis. But for now, maybe just know that aspartate makes oxaloacetate. But you can also get oxaloacetate by pyruvate. So if you have enough pyruvate to make enough acetyl-CoA, you can take some of it and go through pyruvate carboxylase, which you need to know requires biotin to work to make oxaloacetate, and you can also use some of that pyruvate to make acetyl-CoA through pyruvate dehydrogenase complex. So don't confuse pyruvate carboxylase with pyruvate dehydrogenase. X, and then that's just a way to differentiate between the two of them if you do get those confused. But yeah, other than that, I wouldn't really know too much about this because you're going to learn about it later on. And this is your summary slide. Make sure to read those as well. Okay, so your pyruvate dehydrogenase is what allows for the conversion of pyruvate to your acetyl-CoA. So if you have a deficiency in this enzyme, you're going to have a buildup of just pyruvate. And when pyruvate builds up, it's going to have no choice but to undergo turn into lactate through lactate dehydrogenase enzyme. So when that happens, you're going to wind up with chronic lactic acidosis. And that will ultimately lead to neurological defects as well, because you have an accumulation of TCA intermediates in your neurons, and it's just like not good for your neurons. So it's treated with a keto diet, which makes sense. So from this, I would just kind of memorize the bolded words. If you can go back and memorize what you'll see increased serum levels for. But I think the main point is the bolded words. So if he said that somebody suffers from chronic lactic acidosis, right away it's a pyruvate dehydrogenase deficiency. And then, with fumarase deficiency, kind of the same thing. You're going to have neurological impairment. But for this, to differentiate between these two, because they both lead to neurological issues, you can memorize that the urine will contain odd amounts of fumarate and elevated levels of succinate, because your fumarase isn't going to be able to convert fumarate into malate. And so you wind up with a buildup of fumarate and an elevated amount of succinate, which is the precursor to fumarate. So for this is really low yield, just knowing that sometimes if you have these buildup of TCA cycle intermediates, any kind of buildup of literally anything in your cells is very irritating to the cell and can lead to chronic inflammation, and chronic inflammation can lead to cancer. And this is just a summary slide as well, so don't worry too much about it. He's just showing you here where the Krebs cycle occurs, which is in the mitochondrial matrix. And that's also a summary slide because he just wants to show you how everything connects here. So many circles. Okay, that's it. Do you guys want to take a break, or do you want to keep going?
I'm good if you're good. I just had one last question on the pyruvate dehydrogenase complex. Could you just outline really quickly what exactly? I know that on the prosthetic group, or the arm, is that all one arm and it's just moving around, or is there three in that slide? In slide 10?
No, keep asking, sorry.
Yeah, in slide 10, I was just wondering where exactly is the arm, and what is it doing with the acetyl-CoA?
Yeah, so it's just this picture just shows one arm. I'm not sure if the enzyme has multiple arms, but for our purposes, just know it as having one arm of this lipoic acid prosthetic group, and these enzymes work like gears. So you know how one gear rotates.
Up right now, I just see a black screen.
Oh really?
Yeah.
Oops. Sorry.
Oh, you're good.
I thought internet was lagging on my end also, because I thought I was missing something.
No, that's obviously, I think it's my fault. I don't know what's going on today. Giving me so many issues. Can you see it now?
Yeah.
Okay, awesome. Okay, so the pyruvate dehydrogenase complex, they kind of work together as gears. So while this one is turning in one direction, the other one is turning in the other direction. So for the lipoic acid prosthetic arm, it's going to move with the rotation of the enzyme. So while this one rotates, I guess in this direction towards it, it also moves. And when they touch each other, it grabs the pyruvate and brings it with it. Then the CoA group comes in and gets added to the acetyl-CoA. The acetyl-CoA then gets released. And then now your lipoic prosthetic group is just, it has released the acetyl-CoA, so it's just by itself, but it needs to reform that ring that it originally had. So in order to do that, then it kind of touches this gear, which is also rotating, and it, grabs the two hydrogens from it through the FAD group to kind of just, reform that ring. Does that make sense?
Yeah. In the sulfide bonds there, was there something important about that or not?
It's low yield, honestly. It's just showing you that it's reforming the ring, but I don't even remember knowing that last year, so I wouldn't worry about it.
Okay.
Just know that the lipoic prosthetic group is the main group that grabs the pyruvate and does the main reaction.
Okay. Yeah, I see that. Thank you.
If you actually watch he put an animation in the little in the little summary right here that you see in the bottom of the PowerPoint.
Oh, like in the notes section?
Yeah, there's like this animation that it really helped me to understand what was happening. So if you like, if you're like confused about it, definitely check that out because it's actually really helpful.
Oh, thank you for sharing. I didn't even notice that.
Yeah, thanks.
Cool. Any other questions before we move on to the next thing?
Hi, Phoebe. Are there any helpful YouTube videos other than the ones he showed in class that we can kind of get like a summary of all these reactions?
Yeah, I found a few on YouTube. I lost them, but I have them in like my videos last watch. So I'll make sure to post them probably later today if you need them.
Okay, thank you.
You're welcome.
Hey, in addition to that, I think he also posted a couple on the modules. I forgot where exactly, but he posted them by chapter or by topic. And then Gabriella has a question in the chat. Can you see it, Phoebe?
Oh, actually, I can, I think. Can you please re-explain the PDH complex regulation again? I was following along, but then I got lost. Okay, that's the question, right? Or am I like way behind? Okay, thank you. And thank you for sharing that. Oh, it's right here. Okay, so what we decided was true is the acetyl-CoA works both as a, hang on. So it works both as a allosteric regulator to the PDH kinase. So the PDH kinase is what is doing the covalent modifications on the PDH complex. So your PDH is either going to be phosphorylated or dephosphorylated. If it's phosphorylated, it is turned off. If it's dephosphorylated, it is turned on. So acetyl-CoA can go and allosterically upregulate the PDH kinase so that it turns the PDH off by phosphorylation. But it can also act as an allosteric regulator directly onto the PDH complex. So not the PDH kinase, it goes directly to the PDH complex and negatively acts on it. So it inhibits it directly. So it works both roles. And then NADH, ATP, they also do the same thing through the PDH kinase to phosphorylate and turn off the enzyme. And insulin, which wants to promote the TCA cycle and glycolysis and all that, it's going to do the opposite by upregulating the PDH phosphatase, which then removes the phosphate group from PDH and renders it active. Does that answer your question, Gabriella?
Hey, Phoebe.
Yeah.
So when you say it acts negatively on the complex, is it basically doing the same thing, like turning it off?
Yeah, sorry. It turns it off also, just through allosteric means instead of phosphorylation.
Okay, and then the first way, like when it allosterically upregulates the kinase, that's just upregulating the enzyme which phosphorylates it.
Correct, yes.
Okay, thank you.
You're welcome. Okay, any other questions on the TCA cycle? If not, I'm going to start the electron transport chain. Okay. Can you guys see my slide?
Yeah.
Okay, cool. I think it froze. That sucks. Sorry guys, it's like not working. Hang on a second. Finally, okay. Can everybody see my slide now?
Yeah.
Thank you. Okay, so read through the objectives because those are his main points that he wants you to know. And you guys can test yourselves on like how well you know it based on like reading the objectives. Okay, so this is the same slide that he showed like 3 other times, but he kind of just wants to show you that the electron transport chain is going to occur on the inner mitochondrial membrane here. This is another picture of it. And that it's an oxygen-dependent process, like all of the other mitochondrial things, like the Krebs cycle, also oxygen-dependent. So the same slide again. So I would memorize these facts that all of these processes occur in the mitochondria. And then he also kind of introduced the idea of the proton gradient that drives the ATP synthase. So this is kind of a busy slide, but he's just kind of showing you how the TCA cycle, its products, which are its 3 NADH groups, it's 1 FADH and it's 1 GTP that are derived from the TCA cycle, feed into the electron transport chain to act as like energy for it to occur. So what he means by ATP equivalence is that your NADH group, each NADH that gets put into the electron transport system is going to ultimately yield 3 ATP. Each FADH group that is put into the electron transport system will ultimately yield 2 ATP. And then, you know, GTP makes only 1 ATP, which makes sense. So memorize the ATP equivalents for each, because I feel like that was important last year. And, you know, also knowing, you know, how much the TCA cycle. So you can essentially do math to determine like how much ATP you get from the TCA, from one cycle of the TCA cycle based on these ATP equivalents of the electron transport chain. He also kind of introduces the fact that these amino acids and fatty acids also kind of like are involved in the formation of NADH as well, that also go into the electron transport chain to make energy. But you'll learn about that in greater detail later on, but it's just like an introductory slide to that. Yeah. And he repeated this again. So really just wants you to know how much you get from one cycle of the TCA cycle. So 3 NADH, 1 FADH, and 1 GTP. So memorize that.
Doesn't that do 2 turns though?
The TCA cycle?
Yeah, doesn't it spin twice?
Yes. Oh, yeah, you're right, because you're reducing. Okay, yes. So you would do the
would be 6 NADH, 2 FADH2, and 2 GTP.
Yes, but I think that's per turn. So if it turns twice, then you would have, you know, twice as much. Yeah, that makes sense. But yeah, those are the numbers he wants you to know for now. I don't think he's going to make you do math on the exam, because I feel like we were trying to figure out the total amount of ATP for everything, and then he like never asked it. So that was kind of nice, but yeah.
Phoebe, what do you mean by like 2 turns? I was confused with like Lance's question.
So
each glucose molecule makes 2 pyruvate. Each pyruvate goes to 1 acetyl-CoA.
Okay. That makes sense. Sorry.
You're good.
Was that Elizabeth?
Yeah.
Oh, thank you. Okay. This slide, he literally said this again. So memorize these bolded things here as numbers. But basically he like talked a lot on this slide about like heat production and all of this. But really just memorize these key facts and he will not ask about the details. So we know that the mitochondria is important for ATP biosynthesis. And obviously, because we've been talking about that, it also helps with apoptosis mediation, which you'll learn about in pathology, so you don't need to know the details of that for now. Heat production, we literally never learn about, so, you know, just memorize that fact. And then, you know, obviously, intermediately, metabolism, which is what we're talking about now. And then I would definitely know that you get your mitochondria from your mother, regardless of your sex. So, you know, either child is going to get the mitochondria from their mother, boy or girl, it doesn't matter. So that was, I think, a question that he asked on the genetics of getting your mitochondria. So definitely make sure you know that. So this is a high yield slide. So basically, the idea of the electron transport chain is to pump out protons into your intermembrane space. And by pumping out these protons, you're going to have a positive charge on this side, a more negative charge on this side. And as a result of having all those protons, you're also going to have a lowered pH versus the other side, which has a higher pH. Yeah. So, so you start off with your NADH. So this is like your higher energy, it contributes more energy than FADH does. But essentially, your complex 1 is going to oxidize the NADH. It's going to take those electrons from that reaction, and it's going to shuttle them over to coenzyme Q. So then your complex 2, which does not pump out protons, so make sure you know that, it's going to break down FADH2. This is actually part of the TCA cycle, so this is how the electron transport chain kind of directly connects to the TCA cycle. So this is the same FADH that you made in the TCA cycle, but now you're taking those electrons, you're putting them through complex 2, and then you're going to also shuttle it over to coenzyme Q. And this energy allows it to pump out protons. And he said not to really worry about how many protons are pumped for each, because he was like saying that some sources say 2, some sources say 4, but like don't really worry about it because he's not going to ask. But essentially, you are shuttling protons across, and that's what's giving you the energy to pump out the protons. So complex 3 is also going to pump out protons by using these electrons, and they give those electrons to cytochrome C, and cytochrome C will then give those electrons back to complex 4, and complex 4 will use them to help form water from oxygen and protons. So that's why you need oxygen for this process to work. Otherwise, it's completely halted and you can't do anything. So there's a lot of things that you kind of have to know here. You should associate the enzymes with each of the complexes here at the very least. As for these boxed details, they're like lower yield, but I would know which ones use heme and which ones have iron as like an important aspect of each of the complexes. So knowing that complex 1, complex 2, and complex 3 use iron, and then that complex 4 uses copper instead, and that your complex 2, complex 3, and complex 4 use heme. And you don't have to know like the type of heme group or anything like that, but just kind of be wary of like if you were to ask you if you didn't have heme, which complexes would not be able to work, something like that. So yeah, as for this I would say is a little bit lower yield as well, but it's really just showing you like they're what they give it to after they take the electrons. So you can memorize that if you want to, but honestly it's really just knowing the pathway as a whole. So we know that your ATP is directly correlated with the pumping of the protons, because it's going to be the proton gradient that allows for the formation of ATP. So I'll show you in the next slide.
Hey Phoebe, before we go to the next slide.
Yeah.
I have a question. Two things. What does PO means?
Yeah, I was gonna get to that in the next slide.
Right. And then on the bottom, so are those like enzymes, the one that you said kind of don't worry about, the low yield one, the NADH ubiquinone?
Yeah, so I think those are the enzymes that allow for like you've got your NADH dehydrogenase to like pass it over to the coenzyme Q. Like they're like the intermediate enzymes that help with that passing over, because you see that like your succinate or ubiquinone O, it's like you got your succinate here and you got your ubiquinone, which is your Q. I think that's what it is. I don't remember if he said anything about it, but it was low yield my year because I don't even think I even looked at that, to be honest.
Okay, thank you.
One more question. Saren in the chat said that Dr. Sefeng mentioned something about epilepsy and seizures occurring when apoptosis doesn't happen during embryological development. Is this an extra detail he was sharing or something we should know for the test?
Okay, yeah, that's definitely an extra detail. Like he just likes to talk about like these real world examples with all this stuff. But what he's going to ask you are just the things on the slide. So don't worry about like any of the extra things that he says unless he's just talking about what's on the slide. Yeah, so don't worry about the epilepsy and stuff. Okay, so this is what I meant by PO. So this is your phosphate per oxygen ratio. So how many ATP groups you get per use of oxygen. So during one cycle of your electron transport chain, you're going to use up, I guess it's like 1/2 O2 molecule. So that would be just your one oxygen molecule essentially. So that's going to your NADH is going to contribute 3 ATPs per one cycle of your electron transport chain, which is one use of oxygen. Your succinate FADH, which is like together, these are going to only give you 2 per electron transport chain cycle. So the reason for that is because your complex 1 is going to be able to pump out protons, as well as your complex 3 and your complex 4. But your complex 2 does not pump out protons. It just kind of gives its electrons to complex 3 and complex 4. So complex 3 is only 2 ATPs, but when you add complex 1 to it, you're going to get 3 ATPs. So that's why you have 3 for NADH, which is, used in complex 1, versus 2 in your FAD, which only, yields these 2 because it's downstream of your complex 1. But the main idea that he wants you to know is to know what your P to O ratio is. NADH is 3, succinate FADH2 is only 2. And that's why he considers FADH to be lower energy than your NADH, because it only yields 2 ATPs.
So essentially, this is just the ATP equivalent you said in the beginning of the PowerPoint. This is the exact same thing, right?
Yes.
Okay. And we don't have to know numbers of protons or ATP essentially coming from each complex at all? Like, we won't have to calculate that or anything?
No, he's not going to ask you that. He literally just wants you to know these numbers.
That's pretty nice. Awesome.
Yeah. Yeah. His test questions are so nice, even though he's not.
Thanks.
That is hilarious.
Okay, so this is literally just like an introduction. I hate this picture. I completely ignored it my year because it's just so confusing and busy. I like the next picture better. But really he's just introducing cyanide, carbon monoxide, and hydrogen sulfide as things that inhibit complex 4. And then rotenone is, he said that it inhibits somewhere between complex 1 and complex 2. He said that verbally, but in the next slide, he literally just listed it as an inhibitor of complex 1. And that's what I remembered it as last year. So that's what I recommend memorizing. I honestly wouldn't even bother with this whole chart here because it's so confusing and it's just awful. Okay, so really memorizing this is just the main point here. Knowing that complex 1 is going to be inhibited by rotenone, which has the word 1 in it, so that's really easy to remember. Complex 3 is going to be inhibited by antimycin A, and then complex 4 is going to be inhibited by cyanide and carbon monoxide. So this is worse to inhibit your complex 4 because it's the last step in the cycle, and it would just halt everything. And that's why cyanide and carbon monoxide are so deadly versus rotenone, which you can it's not as deadly, at least to us. Is there a question? I think I heard a mic.
If he asks, would he ask questions like, oh, if you halt complex 2, what would happen? Or if you halt complex 3, what would happen? Would he ask those type of questions, or is it more, I don't know, less tertiary? You know what I mean?
Yeah, so if I remember correctly, he literally just asked something like, which complex does rotenone inhibit? He won't ask anything beyond that.
Okay, thank you.
You're welcome. But as a basic thing to understand, it's halting complex 4 is worse than halting complex 1 because at least complex 1, you still have complexes 3 and 4 that can pump things out. And it's at the beginning instead of at the end, which will stop literally everything. Another thing that you should keep kind of understand is that like your iron deficiency anemia. If you don't have iron, because complexes 1, 2, and 3 use iron, they're not going to work as well. So, you can also have issues with your electron transport chain if you have iron deficiency anemia. And that's literally all you have to know for that. It's very straightforward. Yes. Any questions on that? Okay, so he did also introduce the uncoupler here, which is your 2,4-dinitrophenol or DNP. What it does is it's very hydrophobic, so it can just like latch on to a proton and move it across the membrane in either direction. It doesn't matter. And that's what allows for the uncoupling of your electron transport chain and the formation of ATP. So because your electron transport chain is what's causing the proton gradient to be formed, if you mess with that by adding this like a leak, essentially, you're going to inhibit the ability of the ATP to form, or it's not going to be as efficient because now you have a leak. So it kind of uncouples it. So now your ATP production is not directly related to your electron transport chain. So this slide made me want to dye my ear, but I'm going to try to break it down to you as basic as possible. He doesn't ask on this, which was really nice, even though I worked so hard to like learn it. But yeah, he's not going to ask about this. He just wants you to really understand like the what dinitrophenol does. So under normal circumstances, you cannot run your you can't do oxygen consumption, and your electron transport chain isn't really going to work if there's no ADP for the ATP synthase to turn into ATP. So if you add ADP, that allows it to run until the proton gradient is spent, at which point you need to add NADH to turn on this electron transport chain, because that's the first thing that you add to complex 1. And that's what allows it to run. I think somebody's mic is on. And then, so when you add NADH, this will rebuild that proton gradient, and it allows it to run until you run out of ADP, at which point there's no point in making the proton gradient anymore. So you're not going to have oxygen consumption. So remember that oxygen consumption is related to your electron transport chain. So as if your electron transport chain is running, you're going to have oxygen consumption because you're going to be making that water. So when you add ATP again, now your electron transport chain has a purpose and can now rebuild that proton gradient, and ultimately you're going to run out of oxygen and your NADH is used up, so nothing left to do. So that's normal circumstances. So same thing kind of here. If you have your 2,4-dinitrophenol and your NADH is used up. So your NADH is used up, your electron transport chain is not working anymore because now your complexes don't have the energy to do so. You're not going to have the oxygen consumption. When you add your 2,4-dinitrophenol, it's going to allow for the movement of protons across the membrane, and this is going to disrupt the gradient. So as a result, because it disrupts the gradient, your ATP synthase is no longer going to have a proton gradient. So now your ADP, which has not been inhibited or anything, it's just going to build up on the other side. Your ADP, can't be turned into ATP because you don't have a proton gradient anymore. And so it's going to go and upregulate the TCA cycle and glycolysis and all of that. And ultimately, you're going to wind up with an increase in NADH. So now your NADH is turning on your electron transport chain, and your electron transport chain is trying really hard to build up that proton gradient. But because of the leak in the system, because of the DNP, it's not going to be as efficient. So it's just going to keep running and running and running until all the oxygen is consumed. So just kind of notice that oxygen consumption is like directly correlated with your electron transport chain working or running. In the last example, you have the same kind of idea here. You add an oligomycin, which is going to inhibit your ATP synthase. So now, regardless of whether ADP is present or not, your ATP synthase isn't going to work. So your gradient is not going to change, so your electron transport chain is not going to be running because there's no purpose for it to be running. if there's no function for the ATP synthase. So when you add 2,4-dinitrophenol, the dinitrophenol is going to start moving the protons, so you're going to decrease the gradient, and that's going to tell the electron transport chain that now the proton gradient has decreased. That means like, oh, ATP synthase is working, but it's not. So essentially dinitrophenol kind of mimics ATP synthase in the fact that it decreases the proton gradient. So when you add it, it's going to turn on the electron transport system so that the electron transport system can work to rebuild that gradient, and then you'll see the oxygen concentration dropping because you're going to have an increase in oxygen consumption from the electron transport chain being turned on. So did that make sense to everybody? I know this is like the hardest slide on this entire thing.
Yeah, it all made sense, but what was the inhibitor? Was that the oligomycin?
Yeah, it inhibits the ATP synthase.
And that's a secondary response due to DNP being added?
No, so it's like an experiment. So if you were to add oligomycin, the purpose of that is just to turn off ATP synthase so that you can't move the protons to the other side. So that way your proton gradient is already built up. So your electron transport chain just, doesn't work anymore because it's the ATP synthase isn't working, so the proton gradient isn't changing. So when you add the DNP, you're kind of tricking the electron transport chain into thinking that your ATP synthase is working because now you're having a change in the proton gradient. And that allows for the electron transport chain to like turn on and start pumping protons to the other side. Does that make sense?
Yeah, that makes more sense.
Hey, Phoebe, I had a question about the 2,4-DNP. So you were saying that there was like a leak in the membrane. Does that cause a leak in the membrane?
So I just used the, metaphor, a leak. It's like a leak, but what it does is, I described it here. It's the ring that grabs the protons and can like move, because it's so hydrophobic, it can move across the membrane. So like it essentially kind of forms a leak in the sense that it's like a carrier molecule that isn't regulated. It just kind of moves as it wants across the membrane. Does that make sense?
Yeah, that makes more sense. Thank you.
You're welcome. Okay, so pretty much what he wants you to know is that they are coupled, your oxygen consumption and your ATP production. Your oxygen consumption is pretty much correlated to your electron transport chain being turned on, and your ATP production is obviously coupled to your electron transport chain because it relies on that proton gradient. And so if you don't have ADP, there's no reason for your electron transport chain to be turned on because your ATP synthase isn't going to be functioning without the ADP. So you will only have consumption of oxygen, aka electron transport chain, being turned on when ADP is present. And it's correlated to each other. So the amount of ADP added will increase the electron transport chain, which will increase the amount of oxygen consumed depending on the ADP added. So they're like correlated to one another. So that's the main point here. Any other questions on this like really confusing graph?
So Phoebe, when you have what you're trying to say, I'm sorry, I'm just trying to put this together. So what you're trying to say is when you have low ADP concentration, you're going to add this 2,4-DNTP to kind of stimulate the movement of the protons to the intermitochondrial space, and then you're gonna have this oligomycin kind of reduce the gradient to allow the protons to move back into to allow the protons to move to the ATP synthetase to move into to make ATP in the mitochondrial matrix.
So not quite. Sorry if this was super confusing. So these are separate. This is a separate experiment from this experiment here. So they're doing things differently here. So in this example, you're really just like turning off the electron transport system. So then you add the dinitrophenol, which tricks it into thinking that you have, the movement of this gradient. and that movement of the gradient is going to be disrupted. So you're going to have an increase of ADP because your ATP synthase isn't working. So like essentially when you add a leak to the membrane, it messes the gradient up so that your ATP synthase isn't going to be as as functional. So in this example, number 3, it's a separate experiment. So in this experiment, they just wanted to turn ATP synthase off and then add 2,4-dinitrophenol just to see what would happen. So dinitrophenol is just a molecule that tricks your electron transport chain into thinking that your ATP synthase is working. So oligomycin turns ATP synthase off. You add dinitrophenol, and that allows for the movement of protons and triggers the electron transport chain to think that ATP synthase is turned on. So it wants to replenish that proton gradient by pumping more protons through, and that's why you'll see a decrease in oxygen concentration.
Okay, yeah, that makes sense. Thank you.
You're welcome.
And this is for the oxygen concentration inside the matrix.
Yes, correct. Yeah. So it's the oxygen that is being used to make turn into water in complex 4. So I'm going to keep going, but if that's still not clear to you guys, I can go over it again at the end. Okay, so your chemiosmotic theory, you should know what this theory is. I would say that these 4 tenets are very important because you do have to memorize them. So for your chemiosmotic theory, you need to make sure that, so for this whole thing to work, you need to make sure that your inner mitochondrial membrane, which is where the electron transport chain occurs, is impermeable to protons. So the protons, once they're pumped out, they can't go back through just by like going through the membrane because that would ruin everything. The next thing is protons are pumped vectorially across the inner membrane. So, you know that they have to be pumped across the membrane to build up that gradient. And then you, like what we just talked about, is that electron transport and ATP synthesis are tightly coupled. So as you have more electron transport, you have more protons being pumped in the intermembrane space, and you're going to have more a larger gradient that will promote more protons to go through this ATP synthase along their gradient, which allows for the formation of ATP. And then lastly, having the ATP synthase, which forms the ATP. So I would memorize these 4 tenets and understand that the ATP synthase is dependent on this proton motive force. So the proton motive force is based on a difference in chemical potential. So because you have a lot of protons in this intermembrane space, you have a very low pH versus a very high pH in the other side where you have a low amount of protons. Additionally, you have your electrical potential. You have lots of positive charges on this side and lots of negative charges on this side because of the difference in the amount of protons. So this is also going to draw protons into the other side, which allows them to go through the only hole that they can go through, which is this ATP synthase. So it forces them to have to form ATP. So that's the main point here. For this, I personally memorized the subunits, but don't do that because that was just a waste of my time. He didn't ask about it. But I would understand that you've got your F0 subunit, which is embedded in the membrane, and then you've got your head group, your F1 head group, which is going to be on the side of the matrix. And this is the subunit that works on making ATP. So that's what I would know. And then, he showed you that video, which I guess most of us didn't even hear, where, the protons kind of go through and it allows this whole thing to like rotate. And the rotation causes the ADP to kind of come closer to the phosphate group to become ATP. So just having a general idea as to how that works is good enough. And then this is kind of important, but like just knowing a general idea as to how this works is also just, you don't have to know like the nitty-gritty details. So essentially, you're since you're having electrons flow through all of these complexes and you have these carrier molecules that have to grab the electrons, transport them to the next place, and you risk the possibility of these electrons falling off of the complexes and entering the intermembrane space as free electrons, which can react with other molecules and lead to the formation of reactive oxygen species. So in this slide, he kind of gives you a better idea as to how that works. So because your coenzyme Q is shuttling electrons from your first and second complexes to your third complex. Sometimes this coenzyme can accidentally release an electron that will then react with an oxygen in this case and form a superoxide radical. So the superoxide radical will then cause damage unless it's acted upon by superoxide dismutase. And we already talked about these superoxide dismutase and the catalase, I think last block. So you don't have to know the exact reaction, but I would just remember that you have your superoxide radical gets converted into hydrogen peroxide through superoxide dismutase. Then your catalase can come in and turn it into oxygen and water. I would also kind of know that complex 3, because of its association with the coenzyme Q, it's going to cause it has like a major source of ROS production because of this association with it transferring the electrons via coenzyme Q. And then it's this unstable intermediate because it picks up that extra electron that can cause it to react with oxygen, essentially. So that's all I would know for that. Super basic. And then you've got your summary slides. Oh, and then this, don't memorize any of these diseases. Just know that it's maternally inherited. So if he were to ask you, a mitochondrial disorder is, a woman has a mitochondrial disorder, which of her children will have this disease? And the answer would be both children or all of the children, because, you get your mitochondria from your mother regardless of anything. So that's like at most what he would ask for the mitochondrial disorders. And then this slide, like for the millionth time. Okay. Any questions on any of that?
Yeah, Phoebe. So do we need to know really any of those electron carriers like the CoQ and the cytochrome C, or just the CoQ's relation with complex 3? Will that suffice?
So I would know like the order in which they like shuttle between them. So you're like your complex 1 and complex 2 will give electrons to coenzyme Q, which gives it to complex 3, which then gives its electrons to cytochrome C, which then gives its electrons to 4. So just knowing the order is probably enough because he didn't really go into like the details, except for like coenzyme Q with the superoxide radicals. You should also know that fact. But like the teeny tiny, like what it's made up of and all of that, I wouldn't worry too much about.
And Phoebe, just to clarify, for the ROS production, so complex 3 and coenzyme Q both produce the oxide radicals?
Yeah, so from my understanding, because coenzyme Q is giving the electrons to complex 3, I guess like during the transferring process, it can like accidentally like release some of these electrons. So they're kind of both responsible for it. But as a complex, the complex is like the major source because it's like the actual complex instead of like the coenzyme Q, which is just a carrier.
Okay, so I wasn't sure if it was like two separate events that we just needed to know they both did, or if that was like one combined thing.
From my understanding, it's just a one combined thing.
Gotcha.
But yeah, I don't, unless he went into it in detail, but I don't think he did.
Okay. Thank you.
You're welcome.
So the step where CoQ is passing its electrons to complex 3, that's the step where ROS can occur.
Yeah.
Okay.
Because like, if when it's like moving through it, it can just interact with an oxygen that happens to be nearby and just accidentally loses its electrons to the oxygen.
Cool.
So.
Thank you.
Yeah. You're welcome. I do have a Kahoot for you guys. If you guys want to stick around and play, I don't know what time. Oh, we have 9 minutes left. So you guys can leave if you want to, but you can stay and play if you want to also.
Hey Phoebe.
Yeah.
For this slide, for mitochondrial oxygen consumption, so do we just have to know basically what ditrophenol and oligomycin do, like respectively, and not like all the details you gave us?
Yeah, for the most part, I would like if you just know that if you were to turn off ATP synthase, you would still have like a activation of the electron transport chain when you add dinitrophenol. So if you were just to turn off the electron transport system or I'm sorry, turn off the ATP synthase, you would not have activation of the electron transport chain and nothing would happen. But then you add dinitrophenol and it messes with this proton gradient, and just kind of knowing that relationship there is, I think, good enough.
Okay, and then the oligomycin is like the opposite.
Oligomycin turns off ATP synthase and prevents anything from working. So ATP synthase isn't working, the electron transport chain isn't working, so everything's just turned off, nothing happens, there's no oxygen consumption.
Okay, thank you.
You're welcome. Okay, I'm going to start this Kahoot, but if you guys have questions while you're signing up, I can do that. Yeah?
Do we need to know like what the hemes and the iron and like copper does in the complexes?
No, you just have to know that there's iron in there and that there's heme in there. So you don't have to know all the details for that.
Okay, thank you.
You're welcome.
Hey Phoebe, I have a quick question.
Yeah.
So once you add the dinitrophenol, the oxygen consumption starts going down, and that's correlated with the ETC just continuing and turning back on. Is that correct?
Yeah, exactly. Remember that the electron transport chain leads to the direct production of water. So that's going to consume oxygen, and that's what they're referring to. So when you move protons to the other side, the electron transport chain is going to be like, oh, the ATP synthase must be working again. So let's turn on and start, pumping protons and making water.
Okay, that makes sense. Thank you so much.
You're welcome. I'll give you guys like another minute to log in. Okay, I'm going to start it. Okay, so yeah, remember this is pyruvate phosphatase, not pyruvate kinase. Remember insulin is going to upregulate phosphatase, which will activate PDH by removing the phosphate group. So that would be insulin, and then the other three are associated with upregulating pyruvate kinase. Okay, great. Yes, ketone bodies come from acetyl-CoA, but it's pyruvate amino acids and fatty acids that all make acetyl-CoA. Okay, citrate synthase is one of the energy producing steps. The three other enzymes that I put in there are one of those, like, they're all like low yield enzymes for the TCA cycle. So it's the citrate synthase, isocitrate dehydrogenase or whatever, and then the alpha ketoglutarate, which are the three energy producing steps on that one slide where you have like the Gibbs free energy for the TCA cycle.
Hey, Phoebe, on that last question, succinyl-CoA synthetase does create GTP, which is a high energy molecule, while citrate synthase creates citrate from acetyl-CoA. Like, there's no energy produced there. So I'm a little confused on that last question.
Oh, well, he called it energy producing steps. I guess it's because of their very negative low Gibbs free energy. So maybe that's what he was trying to get at. It's a bad question, I guess. So yeah, did I mess that up? Besides that, you said the succinyl-CoA synthetase.
Yeah.
That's what makes GTP. You're right. Yeah. So that was a bad question. I meant it as in like the Gibbs free energy. He called it the energy producing steps for whatever reason. So I was just basing it off of what he said. So yeah, probably should have worded that differently.
I think he was getting at what goes to the TCA cycle and the ETC. So GTP is an energy molecule, but I think he's talking about what leads to making NADH and FADH2, if I'm not mistaken.
Oh, I got you. Yeah, that actually makes way more sense.
You're both right, though.
But citrate synthase doesn't make NADH either.
No, but it leads to it indirectly.
But also you have the what's it called? Is it malate dehydrogenase? It's like further down the citrate acid cycle where he doesn't put it as a free energy step, and it's still producing NADH and the one producing FADH2 as well. So I that was confusing in general because those also give into the ETC.
Okay, yeah, I get what you're all saying. Yeah, that I'm gonna just delete that question and I'll change it to something else after this. But thanks for bringing that to my attention, guys. Because that totally makes sense. Okay, so this question, polysaccharide, obviously sucrose is only a disaccharide, and the others are polysaccharides. So good job, guys. Awesome. Yeah, aldolase is not an irreversible reaction. Those are the three that are highly regulated. By irreversible reaction, he pretty much means that these enzymes can only go in one direction. So when you go through gluconeogenesis, you'll have to use different enzymes in place of these because gluconeogenesis is literally just the opposite of glycolysis. So for these enzymes, you would have to use different enzymes for gluconeogenesis. But yeah, good job. Aldolase is not irreversible. Okay, so for the inhibition of phosphofructokinase, an increased citrate would inhibit it because remember, if you have this increased citrate, you have a lot of other energy producing stuff. So you want to go back and inhibit glycolysis. So it's going to inhibit the phosphofructokinase, whereas fructose 1,6-bisphosphate is not right. It would be fructose 2,6-bisphosphate, but that would upregulate phosphofructokinase. So yeah, that's a trick question, I guess. Awesome. Yeah, complex 2 is like just associated with the membrane. It's not transmembrane, so it's not going to pump out any protons directly. It just helps move the electrons. Awesome. Almost everybody got this one right. Yeah. Maternally, not paternally. Yep, this is just a memorization question. It's, the third complex. And yeah, that's it. So you guys are free to leave. I will see you tomorrow at the TA Q&A sessions. And if anybody, did worse than they were anticipating on the biochem test and need help, please let me know and I will try to help you. If anybody has any comments, concerns, or like want me to do something differently for my TA sessions in the future that may be more helpful, please let me know. Also, I'd like to hear some constructive criticism so I can best work for you guys. I know like the test was a little bit lower in terms of like averages compared to the past years. So I do want to like help you guys out as much as I can. Good job, huh? You did way better than your name would suggest. So that's cool. But yeah, anyways, guys, you are free to leave and stay if you have any other questions.
Hey, Phoebe, I was wondering if when you make questions in the future, could you make them a little bit like the stem clearer? Because I think the confusion on that succinate citrate question was based on whether or not you were asking is if a high energy molecule is involved, like acetyl-CoA is a high energy molecule, versus whether or not it's producing a high energy molecule like NADH, GPTP, those kind of things.
Yeah, okay. I'll try to be more clear in the future. I know like on my practice test there was a couple questions that were kind of really bad. So I'm going to like really revise all of my questions this time because I do kind of want to make it as clear as possible. Because I get that. Yeah. I put that in the Kahoot and I knew it was going to be kind of a confusing question, but I figured since you guys are like my morning session, you're kind of like my guinea pigs for that. And so I like the feedback so I can do a better job at 5 p.m. So sorry about that.
No, thank you.
Oh, you can go first.
I was just saying thank you for the question because I think it will definitely clear up, okay, are we talking about high energy molecules which are involved in the reaction, or is it creating a high energy product? Those kind of things, clarity.
Right. Okay. Yeah. Sorry about that.
I just wanted to say about that question though, Phoebe. I get where you were coming from with that because I had that same doubt when he had that when he mentioned that in the lecture. And so like he specifically used like the same words you used for that question when he was explaining the Gibbs free energy stuff. So I was like trying to understand how he was explaining like why the molecules were high energy or whatever. But yeah, so I get your question. I just don't understand what he was meaning by that during lecture. Maybe I'll just go rewatch it or something, but
yeah, honestly, some of the stuff he says is just so confusing that it just confuses the whole thing. I'm going to rewatch it and get like a for sure answer for you guys because, yeah, it's kind of hard to understand what he means. I just assumed it was like, the free energy of that reaction allows the whole cycle to proceed since, they're so favorable. But I'm going to rewatch that and, double check for what he meant.
So my notes on that slide, just indicate that, he did talk about the free energy, but I think he also was just wanting us to know that the citrate synthase produces the CoA, SH, and then the isocitrate produces NADH, as does the alpha ketoglutarate. And then you also get a CoA and SH from the alpha ketoglutarate. And then he wanted us to know that those are also all regulated in the TCA. So I don't know if he was pointing at like which ones are regulated that produce energy molecules. It's weird. It's definitely because you took it straight from his slides. I get where you were going with it, but it's also like from Seifang.
Yeah, it's confusing.
He's just too smart. He has trouble dumbing things down.
Yes, exactly.
I think it's just regulatory steps. If I think you're right, Lance. I think it's just that's what he was trying to get at. That it's low delta G because it's a regulatory step. But I don't, because it can't be that it feeds into the ETC cycle. It can't be that because there are other steps that produce reduced electron carriers. So I think it's just that. But it was confusing though. I didn't really understand it, to be honest.
Okay, I'm definitely gonna rewatch it. I'll post an announcement based on like what I get from him, just to like clarify what he meant by that, because I don't want to like lead you guys astray. So yeah.
I had a question on glycolysis for the oxidative step. I wasn't sure if that happens with G3P and DHAP, the DAP, or if it just happens with G3P, or like what the order of that was. I keep mixing those up.
Wait, I'm sorry. For what part?
In glycolysis during the oxidative step.
Yeah. So so the oxidative step. So what are you confused about? I'm sorry.
No, you're good. Where the G3P and the DAP, the dihydroxyacetone phosphate, if the oxidative step happens with both of them or just from G3P, or like what the order of that is.
Yeah. Okay. So G3P 2,1,3 biphosphoglycerate is the oxidative step. So it has to go from G3P 2,1,3 biphosphoglycerate. That's the oxidative step. So DHAP, when you break F1,6 biphosphate, fructose 1,6 bisphosphate in half, through aldolase, it's going to make two products: the G3P, which is glucose 3-phosphate, and your DHAP. Your DHAP has to get converted to G3P before it can enter the oxidative step. So it can only enter the oxidative step as your glucose 3-phosphate. So it's an isomerase that will convert DHAP into glucose 3-phosphate.
Okay, so the isomerase switches it to G3P, and that's when it can go through the oxidative step.
Yes, exactly.
Okay, thank you. And then my other quick question was for biotin. Was there somewhere we should know where that came from?
For the what? I'm sorry.
You're good. I don't know if you can hear me well, but from biotin.
Oh, biotin. Oh, you don't have to know where that comes from. It's just like a cofactor that you need in order for that enzyme to work. So just associate, like, if he were to ask you, like, if you don't have biotin, what enzyme won't work? Like, he won't ask it that directly, but, like, just understanding that you need biotin for that enzyme to work, which was the pyruvate carboxylase.
Okay, thank you very much.
You're welcome.
Hey, Phoebe, I actually had a quick question. It's kind of an in-depth question, actually. So I don't know if anyone else has another question, but it's about, I'm kind of confused with antimyosin and how it could be, like, he talked about it being an antibiotic, and I'm confused because I'm assuming we're going over mitochondrial, meaning in eukaryotes. So I guess, like, how could that be an antibiotic if it's also an inhibitor of complex 3, which is in eukaryotes, not prokaryotes?
Yeah, so apparently, I know barbiturates are also an inhibitor of complex 1, and we just take that as a medication. I mean, it's more serious because it is inhibiting complex 3, and that's more downstream. But I'm assuming that, like, as long as you don't overdose on antimyosin, it won't, like, you know, affect us as much. Because a lot of these, like rotenone, I guess because, you know, insects and different types of, not animals, but like, you know, bacteria and stuff, they have like a different type of like energy system. So it's more lethal to them than it is to us. So I'm assuming it has to do with like just having a smaller concentration, which, you know, kind of inhibits our electron transport chain a little bit, but not to like, you know, deadly degrees. Does that make sense?
Yeah, I was just surprised that was, I was more interested that was something we had used as an antibiotic, because I would think that it would have some, like, side effects.
Yeah. I feel like it's like one of those last resort antibiotics.
Yeah. Okay. I was just curious because so as an antibiotic, because I know that like bacterial cells typically use their normal cell membrane for what's it called? The electron transport chain. So is it still like that complex 3 and it shuts down complex 3, or is it do you know?
So it would block complex 3, which would kind of block everything upstream of that, so that only complex 4 can work.
Like in the bacterial cells, is their transport chain the same to like for it to work as an antibiotic? I don't know anything about bacterial.
So yeah, I honestly don't know anything either, but I'm assuming it's because like bacteria have like a slightly different, similar to like how rotenone is like lethal to insects, but like only mildly toxic to us, probably because they just have a different type of, electron transport system such that like the rotenone may, block their complex 4 equivalent or whatever. But I honestly don't know like the details of it. I'm just speculating.
Yeah. I was kind of just thought it was interesting. So
yeah, if you want, you can talk to Safang about it if you're brave enough, I guess.
I don't.
He might like that question though.
No.